This chapter will discuss the interactions between microbes and us humans. Microbes have had a hand in shaping our structure and function because of the close contact that we've had. We've discussed the human microbiome and how this includes all microbes in and on us. They play a major role in our health as well as the general functioning. The majority of the time, our normal flora colonizes us or establishes itself and does not cause disease. Occasionally, a microbe gets past our defense system and can enter healthy tissue. If we see an alteration from health and we see damaged tissues or abnormal functions of organ systems, then we have what's called the disease state. When a microbe causes the disease, it's called an infectious disease. The Human Microbiome Project set out to determine the identity of all microbes on a healthy human. The researchers also wanted to determine how the types of microbes differ in the disease state. They used a lot of technology and genetic sequencing in the process. Our individual cells have roughly 21,000 genes that code for proteins. The microbes in and on us have 8 million protein-coding genes. They also found microbes in areas that they thought were sterile, like the lungs. Researchers also believe there's about 100 million viruses in each gram of human feces. All of us even have a small amount of pathogenic microbes in and on us. Our normal flora in our gut can also play a role in our health. Alterations are linked to mood, heart disease, and diabetes. Areas that have always been known to harbor microbes are the skin, respiratory tract, and GI tract, just to name a few. Some DNA of microbes and/or microbes themselves are believed to be in the lungs, bladder, fetus, and breast milk. DNA of microbes has also been detected in the brain and bloodstream. Our bodies have a large amount of environments that can harbor and support a substantial variety of microbes. The good microbes compete with the bad guys by secreting substances to harm them. Majority of the time, the normal flora is beneficial to us, but can also be of no benefit as in a commensal relationship. There are several factors that have the ability to weaken the host and make them more vulnerable to disease. This includes age, with the elderly and extremely young being most vulnerable. Any genetic abnormalities in the immune system, or AIDS, can weaken the host. Patients undergoing surgery and transplants. Other factors include underlying diseases, medications, lowering the immune system, as well as stress and pregnancy. It wasn't until recently that researchers discovered that the uterus is not sterile like they once believed. Stool samples from newborns prior to their first meal show the presence of bacteria, which indicates their intestines were colonized in the womb. Another route that a newborn is colonized with microbes is through a vaginal birth. The lactobacillus bacteria help the newborn to digest milk, and others have different protective mechanisms. Those born by C-sections have different gut microbiomes initially. A newborn continues to acquire normal flora from breast milk. The gut bacteria help them to digest the sugars. Just to recap how newborns acquire their microbiome. They pick it up in utero, birth, through breast milk, as well as caregivers and their environment. The pathogen is a microbe that takes on a parasitic role and causes infection and disease. Pathogenicity is the ability of a microbe to cause disease. A true pathogen can cause disease in healthy individuals with a normal functioning immune system. An opportunistic pathogen causes disease whenever the host's defenses are down or when it can invade a different body location. Virulence refers to how severe of a disease that a microbe can cause. It's also known as its pathogenicity. It's determined by how well it can establish itself, as well as the ability to cause damage to the host. A virulence factor enhances these abilities. Frequently, when people suffer from infections, it's more than one microbe that causes it, and it's called a polymicrobial infection. For example, the flu can often lead to pneumonia. Skin infections can also be caused by multiple species. Let's start discussing how an organism gets in and causes disease. The first step is that the microbe needs to find a portal of entry. The route can be exogenous, coming from outside of the body. This includes from the environment or another person. The route can also be endogenous, coming from the host itself. Most microbes have found one portal of entry that they prefer, and that is the best habitat for them to survive and replicate. If the pathogen tries to enter the wrong door, they won't cause infection. If the flu virus enters the nasal mucosa, it will cause disease, but if it simply comes into contact with the skin, an infection will not occur. Sometimes microbes like Mycobacterium tuberculosis, Streptococcus, and Staphylococcus can enter more than one portal. The most common portals of entry include the skin, GI tract, the respiratory tract, and the urogenital tract. We will be discussing these in great detail when we get to our disease chapters. Another factor that's important to the course of an infection is the amount of microbes in the inoculating dose. Infection will only occur if a minimum number of microbes or an infectious dose is present. Those that have a smaller infectious dose are more virulent. Every organism is different. Q fever only requires a single cell to cause disease. Tuberculosis has an infectious dose of 10 cells. Gonorrhea has an infectious dose of 1,000 cells, while typhoid fever has an infectious dose of 10,000. Cholera requires 1 billion cells. If the inoculating dose is below the infectious dose, an infection will not occur. The next step after a pathogen finds a portal of entry is to become established in the host by attachment. This depends on receptors on both the host and the pathogen. Organisms use structures like fimbriae and capsules in the attachment process. Attachment is crucial to the organism being able to cause disease, as we have mechanisms for removing them. Bacteria, fungi, and protozoans use fimbriae, proteins, and capsules. Viruses use spikes, and helminths use specialized structures. The next step is that the organism must be able to survive the host defenses. Phagocytes are specialized immune system cells that engulf and destroy pathogens. Most of the time, they are effective at eliminating pathogens. Sometimes, pathogens have antiphagocytic factors. These are virulence factors that the pathogen uses to get away from the phagocytes. The pathogen may release leukocidins that can directly kill the phagocytes. They may produce a slime layer or capsule, and some can even live inside of the phagocyte. The next step is that the pathogen causes disease. There are several different ways to damage the host. A microbe may produce enzymes or toxins. They may also generate an excessively strong immune response from the host or cause epigenetic changes. In A, we can see the secretion of toxins to disrupt the host cell structure and cellular components. In B, we see a pathogen with a capsule who is able to evade the immune system, and then our immune system causes damage to its own cells. In C, we can see alterations to genes of the host. Let's look at different types of enzymes. Exoenzymes can be secreted by the pathogen and cause damage to the host. It breaks down the defenses to allow for further spread. Mucinase digests mucous membranes and is seen in amoebic dysentery. Hyaluronidase breaks down substances that hold cells together. Clostridia and Staphylococci can produce this. Coagulase is produced by pathogenic Staphylococci and clots blood or plasma. Kinase dissolves fibrin clots and speeds up the invasion of damaged tissue. Toxins are poisonous to other organisms. They're named according to their target. For example, neurotoxins target the nervous system. Exotoxins are highly specific and are proteins. They usually target the cell membrane or cause lysis of cells. Chemolysins are exotoxins that rupture blood cells. The image to the right shows the 3 types of hemolytic patterns. Endotoxins are the lipopolysaccharides of gram-negative bacteria. These toxins have a systemic-wide effect. They can lead to fever, inflammation, hemorrhaging, and diarrhea. Blood infections from organisms like Salmonella, Shigella, and others can lead to fatal shock. Let's look at the differences between exotoxins and endotoxins. Exotoxins are toxic in small amounts, while endotoxins are toxic in high doses. Exotoxins exert localized effects on a particular cell, while endotoxins have systemic-wide effects. Exotoxins are made of proteins and are unstable in heat. Endotoxins are made of the LPS and are stable in heat. Our immune system produces antitoxins against exotoxins, but not endotoxins. Exotoxins generally don't cause fever, are released from live cells, and they can be from gram-positive or gram-negative bacteria. Endotoxins cause fever, are released by dying gram-negative bacteria. A sign can be viewed by an observer, such as the presence of bacteria in a sample. A symptom is something the patient feels, like a headache or nausea. A collection of signs and symptoms is called a syndrome. Let's look at the symptoms of inflammation, which is a response from cells and chemicals of our immune system. There's fever, pain, soreness, as well as swelling. The signs of inflammation include edema, which is an accumulation of fluid, granulomas, abscesses, and swollen lymph nodes. How does a physician know there's an infection in the blood? They would look for an increase or decrease in white blood cells. One would also look for septicemia, where microbes are replicating in the blood and in high numbers. Bacteremia and viremia describes a situation where bacteria and viruses are present in the blood, but not replicating. Sometimes infections go undiagnosed because the patient is asymptomatic and does not go to the doctor. The last step for a pathogen is to find a portal of exit. The pathogen is released via secretions, excretions, or discharge. The higher the amount released, the greater chance the microbe has to find another host. Generally, the portal of exit is the same as the entry. Some major portals of exit include skin, sneezing, mosquito bites, needles, feces, and urine. To sum it up, in order for a microbe to cause disease, it must follow these steps. First, find a portal of entry. Next, use fimbriae or other structures to attach to the host. Then it needs to survive the host defenses, release enzymes or toxins to cause disease, and then exit the host. In latent diseases, the pathogen goes dormant and can periodically become active. This occurs in herpes, AIDS, and syphilis, just to name a few. In sequelae, there can be either long-term or permanent damage to tissues. For example, meningitis can lead to deafness, and strep throat can lead to rheumatic heart disease. Let's talk about the course of infection of disease. The first is the incubation period, which is the time from contact with the pathogen to when symptoms arise. Every pathogen and disease differs in their incubation period. It may be several hours to years. Most are 2 to 30 days. The next stage is the prodromal stage, which is a 1 to 2 day period when the earliest symptoms arise. This is generally mild symptoms when one knows they're coming down with something. The acute phase is when the pathogen is replicating at a high rate and we start exhibiting the more severe symptoms. The convalescent period is when our health and strength starts to return to normal. This is usually the time we stop taking antibiotics on our own advice because we think we're better. The continuation phase occurs only in some infections. The patient could be well and still harbor the pathogen, or the organism could be gone, but symptoms persist. Here we see the incubation period, the time of exposure to when we first see symptoms. The prodromal stage is where we exhibit mild symptoms. Our acute phase is where our most severe symptoms are seen. And then finally, the convalescent period, where our health improves. Again, only some infections have the continuation period. Where do pathogens come from? Reservoirs are natural habitats that we would find a pathogen, and that could include us, water, soil, plants, or other animals. A transmitter is how the infection is acquired. Sometimes the reservoir and transmitter are the same, as in syphilis, or they can be different, like in hepatitis A. Living reservoirs can include other animals and arthropods. Let's look at two different human scenarios. An actively ill individual who has the cold or the flu contaminates their pen and then shares it with a classmate. That's an example of transmission through an object. An individual who is a carrier and is not exhibiting symptoms can shed the virus like hepatitis A in his feces and may contaminate food by not washing his hands properly. Living reservoirs include us humans. Carriers harbor a pathogen unknowingly because they don't have symptoms, but can transmit it to others. Asymptomatic carriers show no symptoms, but are infected. Incubating carriers transmit the disease during the incubation period. Convalescent carriers don't have symptoms, but shed the microbe. Chronic carriers harbor the microbe long after recovery and can transmit it. Animals like fleas, mosquitoes, flies, and ticks can also serve as reservoirs. Larger animals, however, can spread the infection. This includes mammals spreading rabies. A biological vector is one who the pathogen uses in its life cycle. A mechanical vector accidentally carries the microbe, like a fly picking up microbes on its legs. Animals can spread their diseases to humans. Rabies can have multiple host involvement, and some have difficult life cycles in the wild. These zoonotic diseases like hantavirus, the plague, and toxoplasmosis have spread because of the close associations we have with animals. They make up about 70% of emerging diseases, and they're difficult to eliminate without completely eliminating the animal reservoir. Non-living reservoirs include soil, water, air, and multiple surfaces. Most of them feed on dead material and cause very little harm. Some can be opportunistic pathogens, and there's always a possibility of picking up these pathogens from things we come into contact with on a daily basis. Communicable diseases can be transmitted from an infected host to another. The transmission can either be direct or indirect. If the disease is easily spread, especially through direct contact like the flu, then it's considered contagious. A non-communicable disease is not infected from the infected host to another. An example of this is an ear infection, which is caused by a person's own microbiota. Transmission involves direct or indirect contact with living or non-living objects. Horizontal transmission is spread in a population. Direct contact arises between people in close contact. Indirect transmission comes from an object like a doorknob. Vector transmission comes from a bite from an arthropod like a mosquito or tick. Vertical transmission is from parent to offspring. Vertical transmission again is from parent to offspring. Horizontal transmission occurs in a population and can arise from direct contact, fomites like a doorknob, air, water, food, or vectors like ticks. Healthcare Healthcare-associated infections, also known as nosocomial infections, are associated with stays in the hospital or a healthcare facility. This occurs in 0.1 to 20% of admitted patients and results in 75,000 deaths a year. Of course, there are factors that make these HAIs unavoidable. The patients are already compromised, trying to fight off the original pathogen, and it's a breeding ground for pathogens. They may acquire infections when surgical procedures allow microbiota to enter tissues. They may also acquire them from equipment, patients, visitors, as well as the medical personnel. The healthcare process may actually increase the chances of transferring pathogens. This includes the reuse of instruments, implanted devices, as well as excessive antimicrobial therapy. The most common HAIs come from 5 major organisms. C. diff causes GI infections, and Staph aureus can cause pneumonia and bloodstream infections. Klebsiella, E. coli, and Enterococcus can all lead to urinary tract infections and surgical site infections. Hospitals employ inspection control officers who implement practices and procedures and is responsible for tracking outbreaks as well as training. It's especially important for nurses and caregivers to receive proper training. Many hospitals have universal precautions. So how do we know what organism is responsible for the disease? Robert Koch developed Koch's postulates, which helps to determine etiological agents of many diseases. The general idea is that you isolate what you think the cause is and inject it into a susceptible population and produce the same effect. Postulate 1 is that you find evidence that a certain microbe is present in every disease case. Postulate 2, you isolate it, cultivate it, and characterize it in the lab. Postulate 3, you inoculate it in a susceptible host and see the same results of the disease. Finally, postulate 4, you re-isolate the same microbe from those subjects. The downfall with Koch's postulates is that not all microbes can be grown in the lab. Also, some viruses may not grow in certain animal subjects, and it's difficult to determine a cause in polymicrobial diseases, which are caused by multiple microbes. Epidemiology focuses on the effects of diseases on a community. It uses multiple fields in the process, including microbiology, anatomy, and statistics. It doesn't just focus on infectious diseases, but also studies diseases like cancer, diabetes, as well as mental illness. A nurse named Florence Nightingale laid the groundwork for epidemiology. She realized that dirt and trash in the war zone were contributing to disease, so she implemented the use of fresh linens and towels for each patient, as well as cleaning. In her records, she showed that more soldiers were dying of diseases than from injuries on the battlefield. The goal of epidemiologists is to identify the causative agents of disease, track behaviors, investigate, and determine sources and modes of transmission. They're also responsible for keeping track of all cases and develop prevention and treatment strategies. Reportable diseases are those that must be reported to health authorities. These reportable diseases include anthrax, botulism, cancer, cholera, West Nile, and gonorrhea, just to name a few. The prevalence of a disease is how many cases are present in a population. To determine prevalence, you would divide the number of cases by the total population. The incidence is the number of new cases over a certain time period. To determine this, you divide the new cases, let's say over 1 year, and divide that by usually 100,000. Mortality rate tells us the number of deaths from a particular disease. The morbidity rate is the number of people with an infectious disease or other disease. The top graph shows the incidence rates or number of new cases of acute hepatitis C from 2000 to 2014 within different age groups. The bottom picture shows us geographical representation of where we have seen Zika cases in the United States. A common source epidemic arises from exposure to a single source of infection, such as meat from a particular plant. A propagated epidemic may remain in a population because it can be transmitted from person to person. A point source epidemic came from a single source and all victims got infected at the same time. An example of this is a contaminated bowl of potato salad at a family barbecue. Figure A shows a common source epidemic curve from an outbreak of Salmonella at a manufacturing plant. B shows a propagated epidemic curve of the flu from fall 2015 to fall 2016. This data gives epidemiologists vital information in their investigations. An index case may or may not be the first case of disease. An endemic disease is one that remains steady in a particular region. Lyme disease is an example of this. A sporadic disease, like tetanus, are reported occasionally at irregular times. An epidemic occurs when higher than normal numbers are present in a population. A pandemic occurs when an epidemic spreads across continents.